Welcome back friends. Myself Dr. Anita Sinha here with you again, and today I am going to discuss transcription initiation process in case of eukaryotes. So let's start with transcription. What is transcription? Transcription is a process through which RNA chain is made along a DNA template. Along a DNA template. And for this synthesis of RNA chain, RNA polymerase II enzyme is mainly involved. So RNA polymerase II enzyme does catalyzes the formation of RNA chain from ribonucleotide triphosphates. Okay. Now, although the process of transcription in case of prokaryote and eukaryote are almost similar, but still in case of eukaryotic transcription the process is somewhat more complex. So before going ahead, I would like to discuss about some of the points which are very important. So first I will discuss about the enzyme RNA polymerase. Second thing I am going to discuss about structure of RNA polymerase II promoters. And third thing I will discuss about various transcription factors which does bind or interact with the promoter region and RNA polymerase II for the initiation of transcription. Process. So first thing here is RNA polymerase. In case of eukaryote, there are 5 different types of RNA polymerases which are involved in catalysis of or in catalyzing the formation of different RNAs. For example, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, RNA polymerase 4 and 5. Okay, let's rub it so that you can get the clear-cut view. So here types of RNA polymerase. Their respective location and their target products. Okay. RNA polymerase type 1. It is located in nucleolus and it is mainly responsible for the synthesis of all types of rRNAs except or excluding 5S rRNA. Next one is RNA polymerase type 2. It is located in nucleus and it is mainly for synthesizing pre-nuclear mRNAs. Okay. Third one, RNA polymerase type 3. It is also found in nucleus and it mainly catalyzes the formation of tRNAs, 5S rRNA as well as snRNAs, small nuclear RNAs. In addition to these three type RNA polymerases, type 1, type 2, type 3, there are two more RNA polymerase, type 4 and RNA polymerase type 5. These are also found in nucleus in case of plant and they mainly synthesizes small interfering RNAs. So this is the types of RNA polymerase, their location and their target product which they catalyzes. So these are the different RNA polymerases along with their functions in case of eukaryote, and in case of prokaryote, RNA polymerases catalyze the formation of RNA chain by themselves only. But here we will see later on that these different RNA polymerases, and especially in case of mRNA synthesis, RNA polymerase 2 does require the help of various transcription factors which we will see in next section. So this is the overall view of the different types of RNA polymerases, their location and their target product. I should draw it. Now, the second point I would like to discuss is about structure of RNA polymerase 2 promoters. Okay. So here the schematic diagram of DNA. And suppose here this is the +1 or designated as +1, or otherwise it is also known as transcription start site. Transcription start site. Okay. So just upstream and the most closest conserved element, or better say module, because this promoter sequence in case of eukaryotes, RNA polymerase 2 promoter, does consist of various conserved elements or modules, better say modules. So So the first conserved element, which is closest to the transcription start site, that is designated as +1. This is the site from where the transcription process does begin, when RNA polymerase recognizes the promoter and binds upon it. So this box is known as Tata box, and it is having its consensus sequence, and the consensus sequence is TA, T, AA, AA. Start location region is -20. So here in this in the middle it will be -30. Okay. Next to the Tata box, there is another conserved element known as GC box, and it is also having its consensus sequence known as triple G, C, double G. Okay, and its location, start location is -40. Now, third conserved element or third conserved module upstream to the GC box is known as cat box, and it is also having its consensus sequence. It's not the enough space, so I am writing here. It is double G, double C, double A, T, C, T. Okay. This is the consensus sequence of cat box. This is here -60 and -80 is there. Next, upstream to this cat box, again GC box is there, another conserved region, conserved element, GC box, and the sequence of GC box here also will be the same: triple G, C, double G, GC box. Okay. And one more conserved element is there, known as octamer box. Octamer box. This may be present or this may not be present. So here what I am telling, this is all about the conserved components or conserved elements or modules of mouse, thymidine, kinase, gene. Okay. Other RNA polymerase II promoter may have these all components or may not have these all components. They can have some of the components. So this is all about the structure of promoter. Now, the third point I am going to discuss is all about various transcription factors and their interaction or their binding to the regions of promoter as well as RNA polymerase 2 so that to form pre-initiation complex. Pre-initiation complex. So, because I have already told that RNA polymerase 2, unlike prokaryotic transcription, unlike prokaryotic RNA polymerase, eukaryotic RNA polymerase does always require the help of various transcription factors such as TF2D, TF2A, TF2B, TF2F, TF2H and TF2E, TF2H and TF2J. Okay, and this is actually I have written in sequence. In sequence. Sequence of what? Sequence with which they does bind to the promoter and RNA polymerase, they interact with each other and they form a pre-initiation complex. So how it is going to be formed? Okay. Let's draw here. Suppose this is the +1 site or the start site. Okay, and here +10, here -10, here -30, here -50 and so on. So at this -30 means at Tata box, TAA. So here the first Bessel transcription factor. Okay, it will be more better if I'll rub it, so the view will be more clear. So suppose this is the +1 site, transcription start site, and this is the +10, -10, -30, -50, etc. So the first transcription factor which does come and bind is TF2D. One thing about TF2D is TF2D does contain Tata binding protein as well as some TBP associated proteins. Okay, so TF2D, which is the first Bessel transcription factor, it comes and binds to the Tata box. TAA, and its complementary will be there. So this is the non-template strand. Because Tata box is having consensus sequence TAA, and it is located on the non-template strand that it generally reads from 5 to 3 prime in the non-template strand. Okay. So it comes and bind TF2D, and this is the Tata binding proteins and Tata binding associated proteins. Okay. Next, after to it, TF2A comes and join the complex. TF2A comes and join the complex, and it followed by TF2B. So first, transcription factor 2D, which contains TBP and TBP associated proteins, it does come and binds to Tata box of the promoter region. Next, TF2A comes and join the complex, followed by the binding of TF2B, and after that TF2F does binds to the or associates with the RNA polymerase 2, TF2F does binds or associate with the RNA polymerase this is RNA polymerase. It first make a complex with RNA polymerase 2 and then it join the complex, and after that TF2E does bind to the downstream of the start site, transcription start site. Okay, so here is the sequence: TF2D, TF2A, TF2B, TF2F plus RNA polymerase 2, TF2E. After this binding, two more transcription factor named as transcription factor 2H and transcription factor 2J does come and bind, but their exact location is not clear. Their exact location where they come and bind is unknown or unclear. But here I have forgotten to tell you one thing, that TF2F is having two subunit. TF2F is having two subunit. TF2F is having two subunit, and one of the subunit is having the activity of unwinding the DNA helix or activity of local unwinding. So most probably it is presumed and most probably TF2F does catalyzes the formation of local segment of unwinded or the local unwinding of DNA strand, and the last two transcription factor which are coming and binding and joining the complex, TF2H is having very important activity known as helicase activity. TF2H is having helicase activity, and it is presumed and most probably it is estimated that this helicase activity of TF2H, it does interact with the RNA polymerase, it travel with the RNA polymerase, and it catalyzes the strand separation, and RNA polymerase will polymerize the RNA strand, or it will synthesize the RNA strand. So this portion of elongation process of the transcription in eukaryote I will be dealing in my next upcoming video. So this is all about the transcription initiation process in eukaryote. So we have seen that how transcription factor 2D does form the assembly and further transcription factors along with RNA polymerase. They interact with RNA polymerase and they formed the pre-initiation complex. So this is all about the transcription initiation process in eukaryote, and the elongation and termination process I'll discuss in my next upcoming videos. So I hope you like this video, and if you like this video, please subscribe my channel and press the bell button and like button and share with your friends. Thank you. Welcome back, friends. Today I'll discuss about the transcription process in eukaryote.